All right, hi everyone. My name is Emily, and as Amber said, I'm part of the picture team. And yes, that is right. Simran is also on the call. So please, if you have any questions while I'm going through things, feel free to put it in the chat and Simran will be able to check those out and answer them as I'm going. Next slide, please. Yeah, so a brief, quick run through of the agenda today. So I'm just going to give a very brief overview of what picture is. Additionally, what it means to contribute to fair science and data management, how picture is fair. And then we will also hear from two of the guest speakers, Andrew St. Martin and Arnaud, with their experiences in using picture to do their research analyses on Biodata Catalyst. Next slide. All right, so to jump into a quick picture overview, next slide. If you're not familiar with picture, picture stands for Patient Information Commons Standard Unification of Research Elements. And Biodata Catalyst, powered by Picture, integrates clinical and genomic data sets across Biodata Catalyst, including TopMed studies, COVID-19 studies, and BioLink data sets. So the Picture UI, or user interface, allows users to search available data using queries to build cohorts. And then the results from these queries can be exported via the API, which is the application programming interface for analysis. And I've included the link to Biodata Catalyst powered by Picture here on the slide. Next slide, please. So here's just a quick summary of the two parts of Picture. There's the UI, user interface, or API, application programming interface. I'm not going to go through this in too much detail. I just wanted to provide it more as a resource for you if you're not familiar. But just to briefly summarize, the user interface provides you with an easy website point and click interface and a query builder tool that can be used to build your query and select your data. And then the data can be downloaded or extracted to an analysis workspace. The same thing can be done in the API, but this actually uses Jupyter notebooks and R and Python code to accomplish the same thing. Next slide, please. So here's what the user interface looks like. So when you go to that link, you'll be able to see the query builder tool where you can search a term of interest. And then you're able to select the study and the category of variables, and then even select the specific variable you're interested in. Next slide. And then once in that variable, you'll be able to filter based on that specific variable. So you can filter your subset of patients using this. And then on the other hand, next slide, please. You can use the API to do this with Python or R code. And so we actually have step-by-step example code that is publicly available on our GitHub repository. And so that is linked here as well. Next slide. All right, so I know last month, the first part of this sort of reproducible science series, they established, the teams at Seven Bridges and Dockstore were talking about fair science. So I quickly just wanted to touch base on this, what it means to manage data in a fair way. Next slide, please. So FAIR stands for findable, accessible, interoperable, and reusable. So how is Picture fair then? Next slide. So Picture allows you to find data because it allows for variable and variant level data search. And then you can export data at the participant level. Next slide. Additionally, Picture is accessible. So one part of Picture that I haven't gone over for time's sake, but open access allows any user, regardless of authorization to access data, to search any clinical variable in Picture, build cohorts, and retrieve aggregate counts. So this means if I'm a researcher and I don't have any access to any dbGaP studies yet, I can go onto Picture Open Access and still be able to search those variables and figure out what data exists. So then I can get a better idea of which data sets to request access to. Next slide. Additionally, Picture is also accessible because all of our API code and examples are publicly available on our GitHub repositories. And Picture is also developed with an open source Apache 2 license. Next slide. So additionally, Picture is interoperable, so you can connect to the Picture API using any workspace of your choice. And you can export and analyze data in either Python or R. Next slide. And so here are two images that show the Picture API examples in both Biodata Catalyst powered by Terra and by 7Bridges. And so here's an example of you have the choice to export it into a workspace that it best fits your research. Next slide. And finally, Picture is reusable. So other users can replicate your cohort creation by building the same query. You can also save and regenerate existing queries, and researchers can run previous code to rebuild cohorts and reproduce analyses. And so this actually goes well into the introduction of our two guest speakers actually. So I believe first up is Andrew St. Martin. I believe he's on the call.
Yep, I'm here.
So I'll go ahead and turn it over to him. Thanks.
Thanks, Emily. Hi, everyone. My name is Andrew St. Martin. I am a statistician, and at the time of this project, I was working for the Center for International Blood and Transplant Research, or CIBMTR, studying different types of bone marrow transplants for hematologic disorders. So today I'm going to focus on our project in the setting of the sickle cell disease with the Biodata Catalyst and Picture. Next slide, please. So to start, I'll just introduce the Secure Sickle Cell Initiative, which was created by the NHLBI and is dedicated to accelerating the development of treatments aimed at genetic-based cures for sickle cell disease. So as most of you or all of you probably know, sickle cell disease is a pretty debilitating hematologic disorder, and it disproportionately affects certain ethnic groups. And so there's quite a lot of interest in studying this disease and looking for cures. In recent years, within the last 20 to 25 years, hematopoietic stem cell transplantation has emerged as a potentially curative modality for sickle cell disease. The CIBMTR collects patient outcome data for sickle cell disease transplant patients, both the pre-transplant information and then periodic long-term follow-up information for patients following transplant. Now, this data is annually uploaded to NHLBI through Biodata Catalysts per this Sickle Cell Initiative program. So every year we submit updated data, including updated patient counts, but also updated follow-up information to Biodata Catalysts for public access. Next slide, please. So the objectives of this project were twofold: primarily to reproduce the results from a recent NHLBI publication, 2019 publication studying the different donor types on sickle cell patients, to show that the data upload from the CIBMTR to NHLBI was seamless and correct, and also to show that the work could be reproduced pretty easily. And second, to perform some novel research on long-term survival for sickle cell disease transplant patients. There was not a lot of research out, current publications about long-term survival, and so there was an interest, and it was suggested this would be a great opportunity to test the Biodata Catalyst and Picture platform to test its ability to perform novel research. Next slide, please. So in brief, the publication we were trying to reproduce the results from was a 2019 publication in Lancet Hematology that looked at just over 900 patients in the United States who received their transplant between 2008 and 2017. And as I mentioned, this was mainly looking at comparing different donor types and their effect on outcomes. So matched sibling being the gold standard donor type that everyone wants to have a matched sibling donor. But not everyone can have, or not everyone has one available. So there are a smattering of alternative donor options, including haplorelatives, and then either matched or mismatched unrelated donors. The median follow-up of this study was relatively short, with 2 to 4 years, depending on the donor group. And that'll play a factor here in a second, and I'll explain that. Ultimately, the study found that recipient age at transplant, the donor type, and conditioning intensity were all found to be significantly associated with the main outcome of interest, which was event-free survival. So either death or transplant failure following transplant. Next slide, please. Now, all 910 of those patients in that publication were included in the annual uploads from the CIBMTR to Biodata Catalyst. So we were able to easily identify and query those patients. There's actually a variable in the database that is a flag variable indicating that those patients were included in this publication. So it was really quite easy to identify them. And then we were able to perform the same analyses that was done for the publication. And you can see that the reproduced results are very similar to the publication results. Now, they're not identical, and this is due to the annual uploads. Not only do they add new patients, but they also add additional follow-up information. So while these patients, from 2019, they had 2 to 4 years of follow-up. When we did this analysis last year, they would have had an additional 2 years of follow-up. So the results are not exactly the same, although you can see that patient age and donor type are still highly significant and significant predictors of event-free survival. The magnitudes are very similar. The effect magnitudes are very similar. And while regimen intensity is no longer a significant predictor of event-free survival, that's not really that surprising, because regimen intensity tends to, have an early effect and then kind of a washout period. So not really that surprising, but we were able to prove that the publication data is able to be reproduced in Biodata Catalyst, and that the data upload from the CIBMTR to Biodata Catalyst was seamless. Next slide, please. So then we embarked on doing some novel research looking at the long-term survival of sickle cell disease patients who receive transplants. And as I mentioned, this is kind of a hot topic because in recent data suggests that adults with sickle cell disease have a life expectancy that is at least 2 decades shorter compared to the general population. And while transplant has emerged as a curative solution, it is associated with some life-threatening complications, most of which occur within 2 years after the transplant. So that begs the question, if patients can survive the early mortality risks, how do they fare long-term? So we updated and expanded our query in Biodata Catalyst to include all sickle cell disease patients who had survived for at least 2 years after transplant. Next slide, please. So we were able to identify in Picture API 950 patients who had received their first transplant for sickle cell disease in the United States, and they ranged all the way back to 2000. So similar characteristics that we would expect to see based on transplant trends, the median age for being around 11 years, same type of distribution for the donor types as that we would expect. And this time we had follow-up of 5 years. The median follow-up was 5 years for the group, but we also had 300 patients in this cohort that had survived for more than 7 years. So we had a pretty decent follow-up in this cohort for looking at long-term survival. Next slide, please. So overall survival for patients who had survived at least 2 years after transplantation was excellent. The probability of surviving for another 5 years, so 7 years after transplant, was 97%. And the probability of surviving for another 8 years or 10-year survival was 96%. For patients who had survived 5 years post-transplant, the additional 5-year survival probability was 98%. Similarly, for patients who had survived for 7 years after transplant, the additional 5-year survival probability was 97%. So really excellent survival probabilities for sickle cell disease patients who can survive past that initial risk period of 2 years. Next slide, please. We also performed a risk factor analysis to look for factors associated with death beyond 2 years, and found that recipient age and donor type is significantly associated with late mortality. Every 10-year increment in patient age at transplant was associated with a 75% increased risk of mortality. And compared to HLA matched sibling transplant, the mortality risk was 3.5 times higher after alternative donor transplantation. We did not see a significant difference in the alternative donors like in the recent publication. So those were just grouped into alternative donor groups. Next slide, please. And the last thing can you go back one? Yeah, thank you. The last thing we wanted to do was, can you go back one slide, please? Was to compare, forward one slide, please. There we go. Okay, this one. We wanted to compare the population to a age, sex, and race matched general US population. And when we do that, we see that the mortality risk of those patients who have survived at least 2 years after transplant was 7 times higher, statistically significant, compared to a general population. The standardized mortality ratio was better for patients who survived 5 and 7 years after transplant, but their risk of death was still 4 and 3 times higher compared to a matched age, sex, and race matched general US population. Last slide, please. So project summary. So we were able to prove that data's upload to Biodata Catalyst is seamless, and that the Picture API really easily enables analyses reproduction of published data. We also showed that the platform allows for wider access to publicly available data and really facilitates novel research nicely. We did all of that work on the novel research in Picture API. That data was actually just recently accepted in a journal last week, so that data has been published or will be published shortly. And it was all done using Biodata Catalyst and Picture, which was really kind of nice. And then in our project summary for the novel research, we found that long-term survival is excellent for sickle cell disease patients who can survive at least 2 years after transplant, despite having a lower life expectancy than that of a age, sex, and race matched population. While this risk recedes over time, it still remains high, even as late as 7 years after transplant. But that's good information that the community wants to know or it needs to know, and we were able to facilitate that using Picture and Biodata Catalyst.
Thank you, Andrew. We did have a question come through in the chat. It says, I would have expected you to be able to go back to the exact version or freeze of the data used in the publication. Can you comment on why that was not feasible?
Yeah, so the data that gets uploaded annually, the follow-up information, it replaces the data that has already been uploaded. I'm not exactly sure why that decision was made to not keep copies of previous uploads, but only the most current data is available in Biodata Catalyst.
And we can open it up for questions for a few moments before we go to the next speaker. So if you have questions for Andrew or about the items that Emily presented on, we have a few moments right now. You can put your questions in the chat or just feel free to unmute yourself. Okay, otherwise I think we will move on to the next speaker. Emily, would you like to introduce them?
Yeah, just real quick, it looks like there was one more question asked.
Yeah, there is.
Yep, was the API used only to obtain the data or for the actual analysis?
No, it was, yep. Yeah, the API was actually used to obtain the data and for the analysis. I should have mentioned that we coded this in R, which is one of the 2 languages available in the API, R and Python. I'm familiar with R, so we use that. And yeah, I was able to build the query and then to perform all the analyses directly in the API.
Awesome. And yeah, so our next speaker is Arnaud, and he will be presenting on his ORCID publication.
Yeah, thank you, Emily. Hi, everybody. So thank you very much for inviting me to present this work. Let me know if you hear me well. And so, yeah, I'm going to present the work that we've been doing with Paula Villac and John Keltman, and that was about reproducing the results of the ORCID clinical trial using biodata catalyst and the picture API. So next slide, please. So the idea was to, general idea was to reproduce the results of this clinical trial and make it, doing so in a very transparent way, streamlining the process of reproducing the results from data access to last outcome analysis. And hopefully strengthen the findings of this clinical trial along the way, as well as starting some supplemental analysis, so some sensitivity analysis and exploratory analysis. And so to show how to do that with a picture and biodata catalyst. So about this clinical trial, the ORCID clinical trial, it has been published on November 2020, and it was a study about the efficacy of hydroxychloroquine on patients hospitalized with COVID-19, and their main outcome was the clinical status at 14 days from inclusion. And so overall, the main finding of this clinical trial was that there was no statistically significant benefit of hydroxychloroquine over a placebo. So control arm was placebo, and hydroxychloroquine did not show any better outcomes in patients hospitalized for COVID-19. Interestingly enough, they use a clinical scale. Not like said, not base their results on biological results that were like very early studies that have been reporting benefits, alleged benefit from hydroxychloroquine used biological results. So this is important to notice. And also we have to notice that results were published in November 2020, which was relatively early in the pandemic. Like it was still like it started to take over the world like around February, March 2020. So from clinical trial perspective, like it's still very, it's a short time to set up a clinical trial, collect the results and publish it a few months. So it's yeah, it was still early in the pandemic, and the question about what treatments works on COVID-19 and whether hydroxychloroquine works was still a very hot one. Next slide, please. So the idea was of the work of reproducing those results was how can we infuse even more confidence and more trust in these results. It was especially important in that context because there has been several treatments that have been proposed at the beginning of the pandemic to treat COVID-19. Hydroxychloroquine was clearly one of them, and there was a lot of polemics around these treatments, whether it works or not, and especially because the claims that it works were based on like clinical trials that were set up very early, and that there was polemic whether the proofs provided by those clinical trials were enough to say that hydroxychloroquine was an efficient treatment. Also, something important to notice is that during COVID-19, there's been numerous publications that have been retracted. So if based on retraction on the website retractionwatch.com, As of February 7, they referenced 69 publications related to COVID-19 that have been retracted, some very mediatic ones, some very important ones have been, like in the Lancet, for instance. So there was clearly some confidence crisis in the results that were published about COVID-19. So it made it even more important to reproduce and to provide very transparent results. So next slide, please. Our method, how we did it. So like the process we are talking about, so streamlining the reproducibility, is based on two major components. So the Biodata Catalyst platform that allows investigators to upload their data and manage access requests. It is especially important in clinical trials data, where obviously there's a huge there's a lot of confidentiality issues. Not everybody can access it. So Biodata Catalyst is a platform that is well suited for to manage access requests, and it provides a single entry point for investigators willing to get access to the data. On the other hand, the Picture API provide easy access to data in a standardized format and through very simple codes using the API. So to be able to, in a notebook, as we did, like in a single self-contained notebook, you can access the data in a few lines and do the analysis subsequently. It is a process of accessing the data from beginning to the end. Next slide, please. The results of this reproduction efforts have been published in JAMIA Open. So if you're interested to see the full details of the study, you can look up there. Next slide, please. Something important to notice about our work is that we focus on the statistical reproducibility. Like there are many issues we can talk about. So how to make biomedical research more reproducible. For instance, for clinical trials, obviously, since a long time now, there is like a very standardized protocol of pre-registration of research hypotheses on clinicaltrials.gov. There is an effort of standardizing the reporting guidelines, accounting for multiple comparisons, and so on. So on our end, this work is more about reproducibility, statistical reproducibility, meaning that if we follow a given protocol, a given clinical trial protocol, and if we get access to like the raw data, do we end up with the same results? It's not, it may sound straightforward, but it's not trivial because there's been some reports that if you like a systematic review from 2014 saying that it's not that uncommon to see discrepancies between what is described in a clinical trial protocol and what is actually reported in the published articles. So the process about data ingestion and data analysis. So first, data has been provided in the form of raw SAS files and different files. So it has been ingested into Picture alongside the data dictionary in a single file. On the other hand, the data analysis code was mostly based on SAS. It was a little bit on it in R, but most of it was SAS. And so we, based on the source code, or the SAS source code, and the clinical trial protocol, we've been we've been writing the analysis code in R and the data management code in R, and the results are being displayed in a Jupyter notebook that is rather user-friendly format that combines the source code and the results, and so it's very easy to see what codes. It makes it more easy to see what code produces what results. And this Jupyter notebook this Jupyter notebook, sorry, because it's one single Jupyter notebook, self-contained, that has been tested on Terra and 7 bridges. So the idea was to allow investigators to be able to run the analysis in one click. There are always, it's hard to maintain compatibility over time in every platform, but that was the goal still. Next slide, please. So these slides present the main results of the original work, so published in JAMA, so the top panel. This bar plot is showing the differences in clinical outcome between the hydroxychloroquine treatment group and placebo group, and basically showing that at 14 days and 28 days, there was no significant differences. And so we've been able to reproduce this figure and the results of the primary outcome within 1 month from the publication of the original clinical trial. And we've been able to display, to have a Jupyter notebook uploaded on GitHub with that results. We kept on working to reproduce the subsequent result, like the secondary analysis and so on, in the following weeks. Although the source code allowed to see the whole process, obviously you still need to get to be granted access to the data to be able to run it, obviously. Next slide, please. So we've been reproducing the main outcomes, also the secondary outcomes. Up to one, everything was similar. Up to one result that was for one of the safety outcomes. Exactly, it was a proportion of patients experiencing cytopenia that was different from what we found. And after reaching out to the authors of the first study, they acknowledged that they actually reported for that one the lymphopenia, and it was just apparently a data management error. So one typo in the data management source code that caused this this very little discrepancy, to be honest. And so besides reproducing the results, we've also been conducting some exploratory analysis and supplemental analysis using the ORCID clinical trial data. What we've done, for example, was to look at the treatments, the co-treatments that were taken by the patients, and treatments that were administered before the inclusion in the clinical trial. And for example, we looked at the azithromycin intake, knowing that azithromycin was one of the treatments that was also in association with hydroxychloroquine, supposedly a potential treatment for COVID-19. So that's why we focused on it. Out of this exploratory analysis, we noticed that patients treated with azithromycin seem to have worse baseline characteristics and overall worst lab status with higher liver enzymes and troponin concentrations. One of the possible interpretations for this could be that patients with more severe condition could be more likely to get off-label drugs, to being given off-label drugs. So that was our main exploratory analysis. Next slide, please. So overall, this process is, I mean, the most important aspect of this whole process is that in the end, there is like a single Jupyter notebook that contains everything from data access to data management to outcomes, reproducibility, outcome analysis in one single notebook, also with the whole code written in R, so accessible by anybody. And this is also displayed publicly on GitHub. So it's an example of how investigators could strengthen their transparency of their analysis, and so build more confidence in the results they publish using some open source tools and using a data repository like Biodata Catalyst. And so in the context of COVID-19, but more in general in biomedical research, this is a very hot topic, like reproducibility of the results. So this is an example of how we can achieve such results. That's all for me. If you have any question.